So I think we'll start now. Hello everyone, thank you for coming to this webinar in which we're talking about the valorization of byproducts for sustainable plant protection. So I will present. Yes, perfect. And do you see my presentation well?
Yes.
Perfect. So this webinar is made in the frame of the FAIRCHAIN project, which is a European project in which it talks about innovative technological, organizational, and social solutions for fairer dairy, fruit, and vegetable value chains. It gathers 25 organizations from 8 different countries in Europe. It started in November 2020 and it ends soon in December 2024. And its purpose is to enable small and mid-sized farmers and food producers to scale up and expand their production of nutritious food through competitive intermediate value chains at local and regional levels. So the aim of the program, it consists in different presentations from around 10 minutes, starting with a global presentation. Oh, sorry, oh, the sound, is it better now? If I talk more closely to...
Yeah.
Okay, perfect. So we'll talk first about a global food processing byproducts and valorization. Then we'll continue with their use in plant protection. And then I'll continue with a focus on basic substances. Then we will talk about whey specifically and vinegar, which are both two plant protection products as basic substances. And we'll finish with a Q&A and a Q&A sessions for 15 minutes, and then I'll conclude this webinar. So I think Mrs. Gorga is here to present
Yes, absolutely.
Our contribution. So I'll let you speak.
Okay. I will have to ask you to move on. So thanks for helping me because I don't have the opportunity to change. So hello, everyone. I am not a partner of the FAIRCHAIN. Yes. Can you hear me? I am not a partner of the FAIRCHAIN project, but I work in France for a Carnot Institute. It's something like a Fraunhofer for the people who are not from France. A Carnot Institute is gathering researchers. So in the agri-food transition, the Carnot Institute, we have 640 researchers that are all active in the food chain. Some are experts of energy, some are experts of upcycling, others are experts of microbiology, but they are all working in the agri-food field, if you want. Next slide, please. You will see all the members of the Agri-Food Transition Carnot Institute. So they are all in western of France, and there are 17 research units. So this was just to present who I work for. Now I will give you some figures about byproducts and the opportunity that byproducts can be for the companies in the food chain. So next slide, please. You can skip next slide, I think. Next one too. I don't want to be too long about the Carnot. And next one, and then we will be there. Okay, so I put there some definitions. Because what is important for the companies is to understand the difference between waste, byproducts. Co-product is a really French term, so sorry about that. So waste, you don't use it, you just discard it. Byproduct, you can valorize it. This is mainly the main difference. Next slide, please, Hortense. Okay. At the Carnot Institute, we have organized a webinar about byproducts because we are talking a lot with the companies, and we have realized that this is an issue. We have in France some national laws that push the professionals to valorize the byproducts, and very often the companies are looking for easy solutions, user-friendly solutions. And we can see, because usually people say that research and business are disconnected. But if you go if you look at the number of publications on the Web of Science about byproducts, you can see that there is high rise in the publication numbers. So that shows the interest and the works that are done on the issue. Next slide, please, Hortense. Okay, so I was talking about the French, the national law that is called Loi AGEC in France. It pushes the reuse and the recovery of byproducts. So there is a very regulatory pressure on the companies to find solutions to valorize their byproducts. Very often, innovation in the food sector is pushed by the regulation. So there is a real issue, and this is why the FAIRCHAIN project is so pertinent. Next slide, please. I have some more figures to share with you. You have here some figures that we have. So all these figures are national. I'm sorry about that, but they are all linked to the French situation. ADEME is the French Agency for Environment, and they have evaluated that each year around 50 million tons of biomass is produced by the agriculture. So this is how much we need to valorize. And in the industry part of the chain, we have 10 to 13 million tons of byproducts produced. So you can see that the first contributor is agriculture, then the milk, the dairy industry, sorry, fruit and vegetables, cereals, and the other ones. So this is the graphic you can see on the bottom right, I think. Yes. Next slide, please. So why there is an interest in the valorization of byproducts? There is the regulation, of course, and then there is the financial issue. Because now recycling costs. Recycling, I mean, like getting rid of byproducts can cost a lot. So recycling is a way to increase your incomes. And there is also an issue about how these byproducts from the agri-food sector can be transformed in added value products, molecules, for cosmetics, for example, for biofuels. So there is a real issue, a real hope to increase the incomes via the use and the valorization of the byproducts. Next slide, please. So here are more key figures. INRAE, that is the French Research Institute on Agri-Food and Environment, has evaluated that byproducts could represent a potential market worth several billions, and this is just in France. The dairy industry is a very good example. The way that is valorized in animal nutrition, for example, represents in 1 billion euros in 2020 in France. So this was a real income for the sector. We have some subsidies and incentives to valorize the byproduct. Horizon 2020 is one of them, and if I am right, I think FAIRCHAIN has been financed within Horizon 2020, or maybe Horizon Europe, I don't know, but one of them. And next slide, please. So this is also French figures, but just to show you. So this is how the byproducts are valorized actually in France. So 77% goes to animal nutrition, and there has been a raise of 43% last 10 years. Then 15% of the byproducts goes to other industries. So it could be food, it could be pharmaceutical industry. Then 6% can be changed into fertilizers, so for agronomic issues. And 1% goes to the energy production. So this is the actual valorization of the byproducts. This will change probably with the research works that will be done in the next years. Next slide, yes. Thanks a lot. So those are the future opportunities. What is really, for now, at the beginning, byproducts were not that interesting, just you could get rid of, it wasn't very expensive. Now you can get some income from the byproducts. Next step is that byproducts are really coherent with the how do you say, with the way people, with like eating local or producing local and or circular economy. So there is a real, it's a clever idea to valorize the byproducts, because there is a real coherence with what society expects from the agri-food sector. So you reduce the food waste, you push the circular economy, and also there is a way, it's a way to enhance and to promote innovation with the development of new molecules and new valorization paths. Next slide, please. So there are some emerging fields. So this is what I was talking about when I was talking about the innovation and the research that could be done. We at the Agri-Food Carnot Institute work a lot on how byproducts could be used as natural fertilizers and natural pesticides to replace the one that we are using now. And there is also a lot of expectations in that field from the consumers. I think that that was my last slide. Can you check if there is another one? Yes. Just those, I will send to Hortense everything, because all the figures that I have put in my presentations comes from these reports. So you will be able to check more if you want. That's all from me, Hortense.
Thank you, Mrs. Gorga, for your presentation. We're a bit earlier because we started earlier, but that's not a problem. And I remind that the presentation is recorded and will be accessible on our YouTube channel. And the slides will be accessible as well on our ITAB website. We will we'll remind you with an email we will send you later. So now I invite Stéphanie Cluzet for her presentation about viticultural byproducts.
Okay, thanks a lot. Is it okay? Do you hear me? Yeah? Okay. So first of all, I would like to thank ITAB for inviting me to present a part of my research work. So I am Stéphanie Cluzet and I am professor at the University of Bordeaux. My research activities are conducted within the Institute of Science of Wine and Vine, and more specifically in the molecules of biological interest axis, which is part of the Enology Joint Research Unit. I will present today the main points obtained from our studies on the valorization of plant-based byproducts for plant protection use, and more particularly, of the potentials of viticultural byproducts. So you can go next slide, please. So we will therefore talk about steel beans. So what are these molecules? They are specialized metabolites from the polyphenol class. In this polyphenol class, you can find flavonoids. For example, you have anthocyanins inside this subclass of polyphenols, and anthocyanins are responsible for the red color, for example, of certain grape berries. Yes, okay. And then you have another subclass of polyphenols, which are non-flavonoids, and steel beans are belonging to this non-flavonoid class. And then I will come back to the structure of these specific molecules, so that characterize them later in my presentation. And then, considering the role of steel beans for plant, in fact, they are considered as phytoalexins. Indeed, these molecules have antimicrobial properties, and their production can be stimulated following an attack by a pathogen, for example, a fungi, like Botrytis cinerea. And so steel beans are major actors in the plant resistance to pathogen. So as I mentioned, the production of these molecules can be induced, so by microorganisms, so biotic stresses, but they can also be induced by abiotic stresses, like, for example, UV light. And these molecules can be also found in a constitutive manner in some plant organs. In addition to their interesting role in plant protection, these molecules are also known for their wide range of biological and pharmacological activities. And so they have a potential valuable health effects. As instance, they have anti-inflammatory, anti-tumoral, antiviral, or even neuroprotective effects. So next slide, please. So let's now look at which plants produce steel beans, as not all botanical families can produce these molecules. This is because the key enzyme involved in the biosynthesis of steel beans compounds, so this is the steel bean synthase, is not found in all plants. So here I am showing you just the botanical families that are high producers of steel beans. So this is Nietacea, Pinacea, Cyperacea, Fabacea, Diptocarpacea, and Vitacea plant families. And hereafter, you can see some examples of agri-food plants containing steel beans. So, for example, you have rhubarb. You can also find pistachio or cocoa and grapevine. So, next slide. So, as I mainly work on grapevine, I will continue on this plant. And in grapevine, the resveratrol is the unit, in fact, the unit of basis for all steel beans. Its skeleton is composed of 2 aromatic cycles and a ethylene bridge. And this skeleton is the one of all steel bean compounds. And the trans-resveratrol molecule is a monomeric steel bean. And this is a molecule which is particularly studied. But in grapevine, you can also find many derivatives of resveratrol. So you can find other monomers, as example, pterostilbene, which is a methylated form of resveratrol. You can also find oligomers of resveratrol, which are more or less complex. For example, you can have dimers like viniferins, or you can find also tetramers, as the one that you can see here, which is a vitizin B, also called R-viniferin. And these steel beans can be found in various parts of grapevine. So, as example, in the grape skin of both red and white cultivars, more particularly when the berries are young berries, or in the green stage of the berries. You can find also these molecules in ruffles of the stems of grapeberry, and also in other parts of grapevine. So next slide, thanks. So the vine shoots or canes, the trunks and the roots are particularly enriched in steel beans. And these organs are grapevine co-products, which are produced each year. So, for example, canes were pruned every year, and trunk and roots can be obtained from annual renewal of vineyard plants. And as you can see at the bottom, it is worth noting that depending on the vine organ that could be considered, and the composition, in fact, of steel beans can vary a lot, both quantitatively, but also qualitatively. For example, in the canes, we are more likely to find monomers and dimers, whereas in the trunk and roots, you will find mainly tetramers and dimers. So next slide, please. So let's now look at the potential application of these steel bean compounds in the fields of agriculture. And given that these molecules have antimicrobial properties, the following question can be raised. So can steel bean extracts represent a green alternative to conventional pesticides? Can we use them as natural pesticides? And so to answer this question, we conducted antimicrobial assays, mainly against 2 pathogens that particularly affect grapevines. These pathogens are Plasmopara viticola. This is the agent, the causal agent of downy mildew, and Botrytis cinerea, which are responsible of grey mold. And firstly, the assays were conducted in the laboratory with canes, trunk, and root extract. And we noted that such steel bean extract that we tested with these two pathogens can inhibited these pathogens with varying degree of protection levels depending on the pathogen and the specific pathogen structure that we consider. So we could have a very high activity against, for example, zoospore mobility, but a less one, a lower one if we consider, for example, the Botrytis mycelial growth. Then we have also tested the steel beans molecules, but in their pure form. So we purify the molecules, and we found that the oligomers, and more particularly the tetramers, were the most active compound. So next slide, please. Then we assess the effectiveness of extract in greenhouse and towards different milieus. So we tested here in this example an extract coming from a grapevine trunk and root parts. We noted that towards grapevine downy mildew, we have a protection around 60%. And towards other mildew that affect, for example, lettuce, cucumber, potato, and tomato, we could have protection between 15 to 80%. So next slide. Then we have also done trials in vineyard to assess the level of protection of some steel bean enriched extract, so in open field. And we observed that when we treated the plants with a high concentration of such extract, we could obtain a protection against downy mildew. But when we used a lower concentration of such extract, we have no protection. So therefore, we questioned the reason for this loss of effectiveness in vineyard. So next slide. And it turns out that some steel beans, not all, but some steel beans can be modified under environmental conditions. This is here the example for transresveratrol. So these molecules can be degraded so by light, particularly by UV rays. And this molecule can be also susceptible to microorganisms, particularly enzymes of some fungi, and for example, Botrytis secreted enzymes. And you have, for example, lacases, which are polyphenol oxidase, who could act and modify the molecules. And we noted that these two environmental factors led to the degradation of 2/3 of the stilbenoids present in our extract. And so next slide. So as a result, we observed a diminution of antimicrobial activities of the extract. And so we are exploring solutions to protect steel beans from these two factors. Currently, we are exploring solution. We are testing the possibility of protecting steel beans against UV by emulsifying the steel beans of extract, or by added flavonoids to the steel beans. So by doing combination steel bean with flavonoids. And regarding fungal enzymes, we are evaluated the potential of tannins coming from other byproducts of grapevine. We evaluated the potential of tannins to inhibit the degradation of steel beans that are caused by lacases, for example. So then next slide, please. And so I thank you for your attention. And if you would like more information, you have here my email address. And so that's all for me. Thanks a lot.
Thank you, Mrs. Cluzet, for your intervention. And now it's my part. I will talk about the valorization of plant protection co-products issued from food valorization, and I focus on the case of basic substances with a definition and a regulatory context. I just introduced myself. I am Hortense Lejeune. I work at ITAB, the French Research Institute for Organic Food and Farming. And what are basic substances? It is a regulatory category of plant protection products. And according to the European regulation, a basic substance is an active substance, which is not a substance of concern, which does not have an inherent capacity to cause endocrine disrupting, neurotoxic, or immunotoxic effects, and is not predominantly used for plant protection, but however has usefulness in plant protection. It is, and the most important point, it is not placed on the market as a plant protection product. So to sum up, a basic substance is a substance that is not primarily intended to be a plant protection product and that has no harmful effect on human or animal health and on the environment. The case of foodstuff. Foodstuff shall also be considered as a basic substance. I will quickly define the word. A food or a foodstuff is any substance or, I think that's better, any substance or product that is expected to be ingested by humans. So food includes drink, chewing gum, and any substance, including water, that is intentionally incorporated into the food during its preparation. However, it does not include feed, live animals, plant prior to harvesting, medicinal products, cosmetics, tobacco, narcotic substances, or residues and contaminants. It can also add additives and flavorings to this definition. In other words, foodstuff, additives, and flavorings can pretend to be basic substances. In terms of the preparation process and the composition of a basic substance, there are 2 points that must be considered for preparation process, which is a basic substance can be used directly in the form in which it is sold, or after a simple transformation process, such as a decoction, dilution, infusion, or infusion in water or in ethanol, for example. However, concerning its composition, it could only exclusively be one or more basic substances. And a basic substance cannot be a combination between one basic substance or more and other substances, such as a coformulant, for example. In that case, the product is considered a plant protection product, which is a specific authorization, which is different from a basic substance authorization. How is a basic substance approved? Well, anyone living in the EU can apply for a basic substance application. It could be a member state or any interested party. And the specificity of a basic substance, it is a substance that is unlimitedly approved, and it has no authorization for selling it and using it for plant protection. Note, however, so the European Commission can freely decide to review the approval of an approved basic substance at any time. So it may lead to the withdrawal of the basic substance or a revision of the conditions of its approval. I will quickly talk about the content of a dossier. The applicant must talk about a general description of the substance, so its identity and its classification and labeling. It has the applicant must provide information about its uses. So a use is a combination between a crop, a mode of application, spraying, foliar spraying, for example, and a pest. And the most important part of the dossier is information about its possible harmful effects, and its impact on human and animal health, about the residues of the substance, its fate and behavior in the environment, and its effects on non-target organisms. Here is the global approval process. I will be quick on that because I'm at the end of my presentation. There are 3 phases. The first case is the admissibility phase, during which the Commission evaluates globally the dossier and its form. In the second time, when the substance is admissible, the Commission asks the EFSA and member states. So EFSA is the European Food Safety Authority, and EFSA and the member state give their scientific opinion. During these 2 phases, questions can be sent to the applicant in order to clarify or complete the file of the basic substance. And once the substance is approved, the Commission published the regulation. It adopts the regulation, and the substance is included in the regulation 540/2011. This process takes approximately 2 years, so it's quite long, and it requires lots of discussion with the applicant. In the second time, when the substance is approved, it is possible for it to be included in the organic regulation. And this whole process is exactly the same for an initial basic substance application or an extension of use of an approved basic substance. So how can basic substances be part of a circular economy approach? I will take...
Stella, sto cercando di mettere questo qui.
I will go quickly on that because I have no more time. So if we consider a grape wine production, it results in production of wine, of grape, and of vinegar. When this vinegar is sent to a food processing company, it uses it for the food production, for food products, for household products, or for plant protection, which interested us. And those products for plant protection made with vinegar are used for disinfecting tools as a basic substance, for a grape wine production, so it closes the circle, or in another production form. Here are all the basic substances that are derived from food processing byproducts. So they are 10 substances out of a total of 27 approved in the EU. As you can see, they are globally all fungicide. And we will, for the rest of our webinar, we will specifically talk about vinegar and whey here. And the last one in gray is the new approved basic substance, which has been approved just 6 days ago. If you want to learn more about basic substances, I invite you to consult our website Substance. You just follow the link here, or you can type substances ITAB, and you will find it soon. So I thank you for your attention, and I let the lead to Hélène Tormeau for her presentation, if she's there.
Yes, I'm here. 2 minutes, please. I have a problem with my computer.
Oh,
Yeah.
okay.
Just few minutes. Okay.
Don't hesitate.
Okay. Okay.
Perfect.
It's okay for me. So thank you, Hortense, to invite me to present my work. It's an opportunity for me and my teams. So I'm from Purpan, Engineer School of Purpan. This school is about agriculture sector engineer, and my specialty is dairy fermented products. So I present here a work of antifungal properties of whey obtained after fermentation of symbiotic culture from Kombucha. Is it a new solution for plant protection? We'll see after. Okay, next slide. So this preliminary work is done by Jessica Caroline Rochas Ribas from Brazil, and she made a PhD, and a part of his research is this field. So as you see before, as you say before, milk and whey exhibit crop protection properties. And whey and cow milk granted of basic substance with antifungic activities according to European legislation for production of vegetable against mildew, yellow leaf curl virus on tomatoes, and agriculture tools disinfection. Proteins and protein hydrolysate was described as the main active component. And perhaps is a component these components of milk produced oxygen radicals in natural light, which may have contributed to the reduction of severity of powdery mildew on treat leaves. So in order to increase these antifungal activities of whey, we tested a fermentation of whey inoculated with SCOBY, used for the elaboration of kombucha, drink made from the fermentation of tea. Kombucha drink is known for these antimicrobial activities, and especially production of acid acetic, phenolic compounds, and protein like as caffeine could have antifungal activities. So we try to combine the two kinds of products to increase the antifungal properties for crop protection. So the objective of this study is to produce and characterize the kombucha-like product from different types of whey and monitoring of fermentation. Second is to characterize antifungal activities of fermentation supernatants against a collection of fungal spoliage strain, and artificial inoculation in pathosystem. Estimation then the type of inhibition, and study the compounds and functional properties of bacteria cellulose produced by fermentation of kombucha. Next slide, please. So whey is the liquid extracted from cheese. You have two kinds of cheese with renate coagulation, and lactic cheese is acid and renate coagulation. These two kinds of cheese. So for extracted from this kind of cheese, we could have sweet whey. At the pH of this whey is 5.7 to 6.5, and acid whey is between 4.2 to 5.6. And for 1 kilogram of cheese, we can have 7 or to 9 liters of whey. Next, yes. So the problem of whey, the whey is a compound very rich in protein and lactose, and this and minerals too. And so the problem is for the same volume, whey has 60 times the organic load of domestic wastewater. So it's problem of pollution, and whey is pollutant, and it's forbidden to release it into the environment, so it needs to be treated. So it could be interesting to have a better add value than waste. So next slide, please. So for this experiment, we made two kinds of two production of kombucha like with whey. So two ACs in November 2023 and April 2024. So the first step is to produce kombucha, and second to elaborate elaborate cheese and extracted whey. So we made kombucha with starter, tea, sugar, and we have a first fermentation lasting 15 days in temperature at 20 to 22 degrees. Then, next slide, please.
Yes, I'm sorry. Could you speed up a bit?
Okay. So in the end of the first fermentation, we take bacterial cellulose, and bacterial cellulose. In this kind of product, we have there is a development of acetic bacteria. So this kind of bacteria produce cellulose to protect them from the sunlight and the oxygen. So this bacterial cellulose is extracted, and SCOBY is symbiotic culture of bacteria and yeast, which is the main microorganism in the liquid of kombucha. So we take both cellulose and 10% of SCOBY to make kombucha with whey and with tea. So there is 5 kind of kombucha: acid whey without pasteurization, acid whey with pasteurization, sweet whey without pasteurization, sweet whey with pasteurization, and tea as a control. And then with this product, we make analysis. Okay, thank you. And fermentation is lasting 21 days in at 22 degrees. So first the objective is to validate the production of kombucha. So as we see in the figure 1 and figure 2, we have the pH deviation between in green D7, not in green, excuse me, in green D21 and D1, in orange D15, 15 days of fermentation compared to mean D1, and in blue D7, the deviation in 7 days of fermentation. So as we see in these two figures, we have acidification, and as we see in this figure, we have better acidification in the test 2, in the second production, than in the first production. The difference could be attributed to the variability of the starter, and the way is done by two different production. So result, we have acidification in the both case. So we evaluate the production of acid acetic by enzymatic kit and by HPLC. So as we see in these figures, we have in the value of acid acetic production. So the production of acid acetic in 21 days of fermentation. So as we've seen, the test for the test 2, we have more acid acetic production than in the test 1. But in both cases, with whey, we have acid acetic production. And for tea, as we see in velvet circle, we have the same quantity, approximately the same quantity in the both test 1 and 2, and is less than whey in the second test. So the conclusion of this measurement, we have growth of acid bacteria in whey, significant growth and significant production of acid acetic in whey, better than tea for test 2. And the concentration of acid acetic is close to vinegar. Just a slide to show you the structure of cellulose, bacterial cellulose. So it's just an example. You have upside in you have the control test is T cellulose, and you have electronic microscopy electronic photos of this cellulose. So you can see acid acetic bacteria in this cellulose. And in the second photography, you have untreated, unpasteurized acid whey. So you see the structure of cellulose is different, and the composition is different. We have more proteins, yellow. You have circle and yellow camembert. Je sais plus quoi, is protein. So you have more protein in cellulose with whey. So the conclusion is that we could make kombucha with whey, and with significant part of cellulose and production of acid acetic bacteria. Next slide. We can Next slide, please. So, evolution for the evolution of the other microorganism, lactic acid bacteria from whey and yeast during the fermentation, the second fermentation. As we can see in the first figure of lactic acid bacteria evolution, we can see that for 3 steps of fermentation, day 1, day 12, and day 21, there is we have survival of lactic acid bacteria during the fermentation, but not in day 21 for both all ways tested and for cheese too. But in tea, there are not lactic acid bacteria, and just few lactic acid bacteria. And in the second figure, you can see the evolution of yeast during the fermentation for the 3 steps of fermentation. So as you see, we have growth of yeast in tea, but not in whey. We have just stabilization of the level of yeast during the fermentation, and in the end, perhaps a decrease of yeast. Okay. So conclude, and in the third figures, we can see the ethanol concentration for all samples. So there is a high level of ethanol concentration in tea in comparison with the others in whey, for example, during day 1, for day 1 and day 21. So in the conclusion, we have growth of acid-acetic bacteria with good activities, significant production of acid acetic, and survival of yeast in whey, and growth in tea, and destruction of viable lactic acid bacteria in the end of the fermentation. Next. So for antifungal susceptibility testing, before the antifungal test, the dry matter content of supernatant was measured and standardized. Supernatant of each samples of fermentation products. Donc briefly, the whey or tea fermented samples were all centrifuged at 7,000 RPM for 15 minutes to remove cell debris. Sterile supernatants were obtained by filtering the supernatants through a sterile microfilter. And to evaluate the effect of fermented whey, to evaluate the effect of whey, tea, and fermented whey or tea on radial mycelia growth, the sample of raw material and the treatments at the beginning and end of the fermentation time were analyzed. So we test eight strains, Aspergillus carbonarius, Aspergillus flavus, Parasiticus, Aspergillus parasiticus, Botrytis cinerare, Colitotrichum misé, Fusarium, Graminarum, and Verticillioides, and Penicillium expansum. All this kind of strain are spoilage and pathogenic fungi from 4 plants. So the sample were mixed with MEA agar at 12%, and the fungal strains tested were deposited in the center of the plates, 10 microliters. The fungal strains were inoculated at 10 million spores per milliliters of the conidial suspension using a molasses counting cell. The plates control.
I'm sorry. Could
yes.
you
yes.
make it
okay.
quick, please?
Okay, so we make control test with tea. And so we have, we can see photographies of the result of the 20 of control And in the end of the fermentation, for the left of on the left on the photography for each columns. So as we can see, we have no growth of fungi for the essay in 21 fermentation, but for whey without fermentation, without the fermentation of kombucha, we didn't have antifungal action. So next, please. So this is, it's not, yes. So just results of antifungal susceptibility testing on different kinds of strain. So we have in the left the test 1 and in the right the test 2. So as you can see, it's a is a radar diagram. So in the radar diagram of the test 1, as we can see, you have a percentage of inhibition less important than in the test 2. And when we have 100%, all the samples, the whey samples in the test 2 are very good activities for antifungal activities, except for Penicillium expansum. We have 16% of inhibition, and Botrytis cinerea 23% of antifungal susceptibility only for pasteurized acid whey. For the other whey, it's okay. We have antifungal activities at 100%. So conclusion: antifungal capacities of fermented tea and whey are very good, especially for the test 2. And perhaps is the importance of the role of acid acetic for whey, the antifungic role of acid acetic, but we don't know now, and we have to characterize all the compounds of kombucha with whey and kombucha with tea. So it's the next slide, the last slide. So conclusion, just we have antifungal properties of fermented whey and fermented tea with kombucha-like production. And now the second step is to have artificial inoculation of plants, banana, and vineyard leaves of vineyard, and cereals and wheat for fusarium. So this is the next step, and we have to resolve the variability of fermentation, of the quality of fermentation of kombucha. So we have to optimize to the process. So thank you for your attention. And yes, next is all the partners of this project. So we have University of Montreal, Laboratoire de Génie Chimique in Toulouse, UMR IAT at Montpellier, and UMR Cali Sud at Montpellier, and the teams of Purpin, all the teams of Purpin. Okay, thank you.
Thank you, Hélène Tormeau, for your presentation. So next one is Clément Gindrat-Keller from Cogiterre and Grangeneuve. And we'll talk about the case study of vinegar. So I invite Mr. Gindrat-Keller to talk. I'm sorry, we're a bit late. So I think the Q&A session will be shorter.
Hello, everybody. You can go on the next slide. So my name is Clément Gindrat. I'm working for Cogiterre. So Cogiterre is the small SME. We are only 5 people, and we are producing vinegar, and we are doing some food consulting for different kind of customer. And we are part of the European project FAIRCHAIN. So next slide. So the European project FAIRCHAIN was done in all Europe. You can see here the map, and I will speak more about the Swiss case study. So in the Swiss case study, there are different kind of partner. There is a Farmer Institute, Grangeneuve, Cogiterre, DSS Plus is a company that is working doing consulting for sustainability project and safety project. There is 2 apple juice producers, so Meul and Biofruit. One is the German part and one is the French part. And there is another consulting company, Food Engineering. So next slide. So in our goal, I will explain a bit what we have done on the FAIRCHAIN project on the fruit and vegetable value chain. So the baseline. When we would like to produce juice, we use apricots or apple and other fruit. We are pressing them, and then in one side we have juice that is going to the consumer, and then we have wet and dry co-product that is going for energy and composting. The goal of FAIRCHAIN was to find new value chain, new innovation for this co-product, for this byproduct. So the baseline is always the same. Apple and apricots are pressed for the juice, and then we have the wet part. So our goal was to use the apple pomace to produce vinegar, and the first goal on the FAIRCHAIN project was to use this vinegar for detergency and phytosanitary application. The idea was a bit the same as we've seen before, is that the juice producer can produce that as their own detergency agent or phytosanitary agent. For the dry bio co-product, the apricot pits was used to produce biochar, and the goal of the biochar is the soil health and to capture carbon. After that, the goal of FAIRCHAIN project was not only to focus on the scientific study, but to have a business model that can work on in Switzerland and in Europe. So next slide. So just a bit more to explain what we have done. So apple trees are coming directly from the tree. They are sorting, and then they have a press. After the press, as I've said before, we have the juice and we have the apple pomace. But the first goal is we would like to have a fermentation with vinegar with the apple pomace. I've tried to do some solid state fermentation with apple pomace, so it means that only yeast and apple pomace, and it didn't work. I have a lot of malt. So the goal was more to control this process, and for that we have to dilute the apple pomace with water, but then we are going to dilute to the amount of sugar. So we were looking for another co-product, and the other co-product that we found, it's molasses, and molasses, it's a co-product from the sugar industry. Then we mix these 3 different ingredients, and we had then we can say an apple pomace drink. We were looking too for another source of sugar. We were looking for potatoes or different kind of cereals. But at the end, we were only working with water, molasses, and sugar. After we made the first fermentation, and our goal was to go until 8% of ethanol inside, and the first fermentation was really quite well, and we have an apple pomace wine. So it's not because we had molasses inside. Is not a really a good wine. It's quite if you drink it, it's quite disgusting. But the first goal was not to go in human consumption. Then what we have to do a decantation to have the liquid part, and the liquid part will be transformed into vinegar, and we have the solid part. During the first FAIRCHAIN project, we have met a startup called Low Impact Food, and they are working with insect. And they see the goal of this insect is to go on human consumption, and they were really interested in our fermented apple pomace, so the solid part after decantation. This apple pomace had helped the growth of these insects. They had a better yield, and they have a more stable reproduction. So with the liquid part, we've made an acidification with acetic acid bacteria, but we were not working with a specific strain of acid bacteria. We were working with I think we said vinegar, like a SCOBY, but vinegar mother, just to start the fermentation. And at the end, we have a vinegar. The goal of this vinegar was to be used in the detergency application. So INRAE have studied that in Lille, and Grangeneuve have studied the phytosanitary application. Then we had another contact with different clients that are using our vinegar for animal feeding, and we are in contact with different kind of biochemistry detergency agent company that would like to do some soap for hand or different kind of application. Next slide. So here is just a small picture of that's what we have done. You can see the dry product and the wet product. The second picture is it's the soaps that we have. So it means the apple pomace wine, and in the third picture, it's the vinegar, the application that we had on the field for biostimulation or for the apple tree. Next slide. So what have I won't speak about all studies, so it will take a couple of hours. But just one study that I've made in INRAE. INRAE was studied the disinfection of our vinegar in different kind of bacteria or spores, and here it's just microscopic picture, the comparison of effect of our vinegar on Saccharomyces cerevisiae. Why we are working on this yeast is this yeast don't put directly food safety problem, but if we have Saccharomyces in food industry, it will transform the sugar into ethanol, and then it will have ethanol in soft drinks, and that could be quite dangerous for the kids or another kind of people. So you can see here that the FAIRCHAIN vinegar FCV has quite an effect to for the detergency effect to clean stainless surfaces. We have first picture is untreated stainless. Second one is with water, and third is with acetic acid, only acetic acid 5% and acetic acid 20%, and we compare it to NaOH 0.5% because it's what is used in industry to clean the CIP. So we can see that the FAIRCHAIN vinegar has an effect on Saccharomyces cerevisiae. Next slide. So what we've made with this technological and organization innovation, we have produced a sustainable acetic biomass extract. So we've made some vinegar from co-product. We have produced a biosourced vinegar detergent. One of our customers now is using this vinegar to clean the vertical farming. installation that they used to where salad or are growing now. What was quite interesting, it's not only the scientist view, but now the problematic that have the juice fruit producer is that they have to pay for the supply chain cost to throw away the apple pomacy. And with this project, now we can change this paradigm. With our vinegar that we produce, we can pay this co-product, so it so the apple juice producer will get some money for that before they have to pay for it. Of course, we can pay ourselves for the transformation from byproduct into vinegar, and this product is a bit cheaper as we can find on the market. So that is quite interesting. So we made a production of vinegar from apple pomacy, and we have the possibility to have a biosourced detergent. So thank you, and thank you for listening. My presentation is over.
Thank you very much. Thank you. Well, then the second case about vinegar also is from the company Charbonneaux Brabant, in the contact of Mrs. Delima-Nicolay, which cannot be there today, so I will present this presentation in her behalf. I hope she will be satisfied by my presentation. And I'll make it quick because we're a bit late. So about the company, Charbonneaux Brabant is the first French producer of vinegar. And how is their vinegar valorized? It is valorized in the food sector, in cleaning and disinfection products, and in the plant protection sector, but that was not initially the case. They have two main activities. The first is the production of vinegar with wine vinegar, alcohol vinegar, and balsamic vinegar. And the second is through their trade and chemistry department, the formulation of detergent and vinegar-based products for the supermarkets. So Charbonneaux Brabant is responsible for the approval of two extensions of use for the basic substance vinegar. And why is it? It was a good opportunity for Charbonneaux Brabant to make those extensions of use, because there were a lot of confusion between vinegar and acetic acid, which is an active substance used in biocontrol products, and it was a relevant way to protect and promote natural vinegar and distinguish it from the acetic acid. So they met from their feedback about the process, about their first extension of use. It was for them, they were given a strong methodological support by us, ITAB, and the uses approved were the weeding of aromatic herbs, so it is not a use for organic production. And their feedback about their second extension of use, it's that it was important to distinguish vinegar from acetic acid in order to make it approve and to approve more uses. It was a large work for the company to construct the arguments, especially because of the regulation aspects. And the uses approved through this second extension of use is also weeding, but in public spaces, so in parks, roads, and walkways. It took for them about 3 years to approve those two extensions of use, so it was longer than the global time for a basic substance application. And what were their global difficulties? Well, I talked about it sooner. It was to distinguish acetic acid and vinegar. Also, acetic acid is a biocontrol substance, so it was challenging for them to obtain more than that was already permitted for acetic acid for biocontrol. And it was difficult to be heard in the world of chemistry because it was a large because large organizations didn't really like this kind of approval. And how was the vinegar integrated into their production? So for their production, there was no need to implement a specific process because the vinegar is already meets the food grade specifications. And for the marketing, there were adjustments that needs to be met to require both sales and channels for vinegar as a basic substance. So for the industrial clients, they would buy directly vinegar to sell their own basic substance-based products. There was no specific change. However, for the supermarkets and local authorities, they needed to add a label basic substance with specifying the regulation on detergent products. So how was those process useful for the company? At the time, it was not so. The activity was not so intense. However, now it is. So there's a growing demand for the vinegar, as it takes time for this type of use to make root among the public and the industry. It hasn't revolutionized the activity of the company. However, there's a significance for highlighting the vinegar designation contrary to the acetic acid. It brought value to the company and also other vinegar producers, and it promotes a more sustainable solution that has proven itself and that needed to be democratized. So their feedback is that there's a desire to push things forward and make things progress. If you have some questions for the company Charbonneaux Brabant, you can send directly an email to Mrs. Delima-Nicolay. Here is her email address. So we are a bit late, but if you have some questions, you can write them in the chat, and we will answer. Me and the other intervenants will answer them. So I think there is one question for me about the basic substance process. At the end of the day, the approval processes of basic substance extension for plant protection and chemicals active substance seems quite similar, at least in terms of step times data requirement, isn't it? Well, that's the global information is the same. However, the requirements are a bit less for basic substances than for plant protection products. Also, the approval of an active substance is it is really expensive compared to a basic substance because every applicant has to pay a fee to the European Commission. However, for a basic substance, it is not the case. So it is the main difference between those two type of substances. Don't hesitate if you have any questions to put them on the chat. Well, I guess there is no more question. So to conclude, what I can tell is that there is an economical and an environmental interest in valorizing byproducts from the food industry. And it is a great way it is important to develop value chains for plant protection. And I just noted the importance of, in terms of volumes of byproducts to valorize, because there are only 6% of them that are used for agronomic purposes. And in the case of whey, it has pollution issues too, so it is important to valorize whey specifically. Globally, we can tell that there's a positive feedback of the actual of what is actually made for valorizing those byproducts for plant protection specifically, or even I heard biostimulation, which could be another subject and could be the aim of another webinar. And I have some information for you. The slides will be accessible on our website, itab.bio, and the replay will be accessible on our ITAB YouTube channel in January. We will send you an email with the different links for the slides and the replay. If you want to learn more about the FAIRCHAIN project, I invite you to consult the FAIRCHAIN link, which is this. And if you want to learn more about basic substances, you can check our substance website. It is in French, however, the information in the website are also for the European scale. So thank you very much for listening to our all presentations, and I hope you have learned something. I hope the valorization of byproducts will be more and more in the time. There will be more and more actions in the time. And if you have any words to say, I will thank all the participants and all the contributors. Thank you very much. I will thank my colleagues for helping me for this webinar, and I will thank specifically my colleague Mathilde Renard, who entirely organized this webinar and couldn't be present to animate it, so I animated it for her. So thank you.
Thank you very much.
Thanks to you, and have a nice weekend.
Yes,
Bye.
you too. Bye.
Thank you. Bye.